Hello students, this is a new tutorial on the chapter Principles of Inheritance and Variations. This chapter carries 5 marks and it is there for the first formative exam. This chapter is also known by the name Heredity and Variation. Okay, so let's take the first slide. Now in this chapter we are going to discuss a lot about genetics. So, what exactly do we need to know in genetics? The first thing that we need to know is that organisms, when they reproduce, their offsprings resemble the organism or resembles the adult. So, in these lovely pictures that you can see here, you can see that the young one of a lion resembles the lion, the young one of a rabbit resembles the rabbit. So, there is resemblance to the parent. Yet at the same time, there is no total resemblance to the parent. So, the branch of biology that deals with the inheritance and variation is termed as genetics. So, as you can see, genetics involves the study of inheritance, that is, the process by which characters are passed from parent to the progeny or the offspring, that is inheritance. And the second thing that genetics involves is variation. So what is variation? It is the degree by which progeny differ from their parents. So please keep in mind, genetics involves resemblances because of inheritance and differences among the parents and their offsprings because of variation. So this slide here is showing you how different human families, you will see that you can identify their offsprings because they will have some traits in common. Okay, so you can have a look at these pictures here showing you families and their offsprings and the resemblance and the differences between parents and offsprings. Coming to the history, the fact that variation is due to sexual reproduction is not a new finding. It was known much before, that is around 8000 to 1000 BC. So it's not a new finding. And this finding that in sexual reproduction there is variation, this fact is exploited in breeding of organisms, be it plants or animals. So, you might have heard of artificial selection and domestication in breeding of animals. Like, for example, the wild cow Sahiwal. Okay, so this is a superior breed in animals, and it has been brought about by exploitation of this fact that through sexual reproduction variations can be brought about, can be integrated, and humans have the capacity to choose and breed the high quality animal or plants. So we say in case of plants, high yielding plants; in case of animals, we say superior breeding animals. Some of the terminologies you'll come across in this chapter that you need to learn. Okay, genetics, which we already talked about, that is, it deals with heredity and variation. Heredity. What is heredity? It is the transmission of characters from parents to their offsprings. What is variation? Variation is the difference among offsprings. This is already discussed. Hereditary variations. Please note the variations which are genetic and inheritable are called as hereditary variations. Now, same way we have environmental variations. The difference between these two are that environmental variations are not inherited. They are not got from your parents, but these are the variations that you acquire in your lifetime from the environment that you grow up in. Now, So this was the introductory part of the chapter. Now we're coming to some of the very important concepts. And the first thing is, who was the person who is considered as the father of modern genetics? So it is Gregor Mendel, who gave the principles of heredity. And today we have different laws which we call as the principles of heredity because of this man. So it all started with him and when he studied the pea plants. So we will see his experiments on the pea plants, and during his lifetime his work was not recognized, but much later, and especially in present day, his findings have become very significant in the field of genetics. So this is to be kept in mind. Now, what were some of the things that Mendel did? Okay, what were Mendel's findings? What were his conclusions? What was his approach? So he conducted hybridization experiments with pea plants. Hybridization, as you know, is a kind of cross pollination, and accordingly, he proposed the different laws or principles of inheritance that we are going to study in great detail in this chapter. He is also said to have used statistics and mathematics to explain biological concepts and to clarify biological problems. He's said to be the first person to do so. So he has used a very integrative approach using statistics, mathematics, and a life science branch, that is biology. Then he has based all his findings using large samples. So there is great credibility to his data. It's not that he has proposed these laws of inheritance just by doing a few simple experiments, but it was a body of 7 years of work. So there's a lot of credibility to his data. Then one very important thing that he did was whenever he uh, conducted experiments with his pea plants, he made sure they are true breeding. What is the meaning of this word true breeding? For example, if he was selecting a tall pea plant, he ensured that this pea plant, okay, self pollinated many times, and it in every generation this pea plant produced only tall plants. It never produced dwarf plants. So he ensured that the plant that he is using for his experiments is pure or true breeding. Okay, then he confirmed all his inferences. So this is another thing he did, and he usually conducted experiments using plants of opposing traits. So one clear example here is if he used a tall pea plant, then he crossed it with a dwarf pea plant. So you can see the alternating forms of the trait or the opposing forms of the trait. Or if he used a pea plant with yellow seeds, he crossed it with a plant with green seeds. So this is what he did with his experiment. So we are going to now take a look at his work. Okay, so before that, we have to know why he selected P for his experiments and which were the 7 pairs of contrasting characters that he conducted experiments with. So the different traits in P that he selected were height, seed shape, seed color. Now this is not mentioned in your book. Okay, so I will also attach in Google Classrooms a corrected list. It's not that it is wrong. It's some books give it differently. In your textbook, it is given as flower color. In this presentation, it was given as seed coat color. So there is a difference in what different books give. But he considered 7 traits, and in your textbook, it is said height, seed shape, seed color, flower color, pod shape, pod color, and flower position. So only this thing is different in your text. So I will be giving you the corrected sheet. Okay, in Google Classrooms. So now let's take a look at the alternative forms of the trait that he selected. So if he selected pea plants based on their height, he worked with two alternating forms of that trait height. So which were the two alternating forms? Tall and short. If he selected the trait seed shape, again he worked with two alternating forms, those which had round seed shape and those which had wrinkled seed shape. Are you getting what it is? So he selected these different traits, and for each of these different traits he selected alternating forms. So if I ask you the question, pod color, what were the alternating forms for the trait pod color? So your answer would be green pod colored plants and yellow pod colored plants. Now, in these two alternating forms, okay, one of the form was a dominant trait. Very soon I'll be explaining to you what's the meaning of a dominant trait, and the other was a recessive trait. Dominant trait, from the name itself, it comes to know you come to know that it is something that is expressed, whereas recessive trait is something that is hidden, because the dominant trait does not allow it to express. Okay, so these two things are to be kept in mind. Dominant means expressed, recessive means hidden. So I hope you've understood the seven traits that he chose and the alternating forms of the traits. Clear? Then again, we need to familiarize ourselves with certain terms. So we will be coming across these terms in the next slide. So I want you to remember these terms. First is phenotype. So the best way to explain phenotype is, if we go back to the slide, if I ask you this particular plant, what is the phenotype? You will say it is the characteristic of tall. What is the phenotype of this plant? It is short. Now, suppose I ask you, what is the genotype of this plant? Genotype means what are the genes that give rise to this characteristic of tall. Okay, so that you cannot see by looking at the plant. So please remember that phenotype is the external appearance. I can give you another example. In human beings, there are people with blue eyes or brown eyes or black eyes. So that eye color, what you see, is the trait or the expressed form. But that eye color or of blue eyes or black eyes is brought about because that person has a certain set of genes, that is called its genotype, which you cannot see, but it is phenotype is a result of the expression of the genotype. I hope I'm clear with these two terms. So this is observable, this is the gene genes which are contributing to this phenotype. So this is not observable. Genotype is not observable. Now we'll also come across the term phenotypic ratio and genotypic ratio, which you will get to know a little later. So it's the correct proportion of phenotypes in a population, correct proportion of genotypes in the population. Just remember these two things at this stage. Now this is very important. Now, an individual, any individual, can either be a homozygous individual or a heterozygous individual depending on the genotype. So, earlier I was talking about this term genotype, which we cannot see, which expresses the phenotype. So, this is exactly what is the genotype. So, the genotype of a person is the genes present which cause the phenotype. So, here in this genotype, for example, capital T, this genotype is going to express the characteristic of a tall plant. Okay, so since both these, capital T is to show you a dominant gene, okay, and small t is to show you a recessive gene. So this particular individual, if you see this pea plant, which has the genotype of capital T, you will see that the phenotype of this pea plant is tall. Why tall? Because it has both dominant genes. Okay, does that mean all tall pea plants have to be having the genotype capital T? The answer is no. These are only the homozygous tall plants. Now there's another set of plants called as heterozygous tall plants. Now heterozygous tall plants are also tall plants because they have the dominant allele or gene capital T. Okay, the only thing is that both these alleles or both these genes in this pair are not the same. That's why they are called as heterozygous tall. Okay, so it is tall because it has the dominant allele, but it is heterozygous because there are two different alleles or forms of a gene in this gene pair. Whereas if you see this individual, this we will call as homozygous but dwarf. Why? Because it has both recessive alleles. So just to make it very clear again over here, because this homozygous heterozygous will be used very often in this chapter, capital T refers to a homozygous dominant individual. Okay, so this is a tall pea plant which is homozygous, whereas this is a heterozygous tall pea plant because you have two different genes in the pair. And this again is a homozygous dwarf or short pea plant because it has small t, small t. I hope this is clear. Now, in the next slide, again, we come across this. I already told you that a dominant gene will show its characteristic or trait, and a recessive gene will hide its characteristic or trait. So going back to this slide where we said dominant traits, tall is brought about by a dominant gene, capital T, whereas dwarf pea plant is because it has the recessive gene, small t. Round is a dominant trait brought by capital R, whereas wrinkled seeds by a recessive gene, small r. Similarly, whatever is dominant, you will write the capital sign. So for yellow, the gene will be capital Y. For green, since it's recessive, it will be small y. Inflated, capital I. Constricted, which is recessive trait, will be small I. Okay, I hope this is clear. So this is the dominant gene which causes the expression of dominant character, recessive gene which fails to express. Hybrid, already you know, it is a plant that you get by crossing two parents. So a hybrid pea plant is you cross two pea, the male and the female parent, and you will get a offspring which is a hybrid. Now back cross and test cross will be doing a little later, so we'll just skip that for the moment. Then this is important. You can get a question asking you why Mendel specifically chose the pea plant. So he had various reasons. The first reason here is pure variety of pea plants are available. Pea plants are very easy to cultivate. The life cycle of the plants only for few months. Contrasting traits are observed. So as you saw, he got 7 traits, and each of the trait had contrasting forms. Flowers are bisexually, so normally they are self-pollinating. But if you want to cross-pollinate them easily, you can do it manually. Okay, by um, emasculation method, okay, of dusting the pollen on that specific flower. So you don't allow self pollination to take place, but you dust your selected pollen on the stigmas of selected flowers, and you can bring about cross pollination. And hybrids are fertile. So some of the reasons why Mendel selected pea plants for his experiments. Now, what are his findings? The first thing that he shed a lot of light on was inheritance of one gene. Now, to understand this, we need to understand this term monohybrid cross. So he conducted a lot of monohybrid cross pollinations. That means a, a, a cross between two parents, okay, based on one trait. Okay, so one trait and two contrasting forms. So I'm going to show you an example of a monohybrid cross, which is very simple. Okay, and what he first we will see what he got when he did a monohybrid cross, and then we'll go to the example. So, like I told you, he explained inheritance of one gene by doing monohybrid crosses. So, what he noticed was when he crossed, for example, a tall parent with a dwarf parent, okay, he got the first generation of offsprings, which he called as the F1 generation or the filial one generation or the first hybrid generation. Now, these F1 offsprings that he got, he noticed resembled only one of the two parents. And in this case, when he crossed a tall parent and the dwarf parent, he noticed that the F1 plant offsprings were all tall and none of them were dwarf. And next, what he did was that he self pollinated the F1 offspring to get the second generation. So, in the first case, what he did was he crossed two parents to get the F1 generation, which he noticed resembled only one of the two parents. Okay, and in this example, all were tall. In the second case, he took all these tall F1 plants and he self pollinated them and he got the F2 generation. Now he was surprised because in the F2 generation he noticed both the traits, the dwarf trait and the tall trait. So F2 plants or the F2 generation or the filial 2 generation showed both the traits. But what did we say about the F1 generation? It showed only one of the two traits. So here both tall and dwarf were seen, but the ratio was different. So he noticed that if from say for example, if he got 100 plants in the F2 generation, 25 plants were dwarf or recessive and 75 plants were tall. Okay, so that works out to a 3:1 ratio. That means 3, okay, out of the 4 plants were tall or dominant, and 1 out of the 4 plants, okay, were dwarf. I hope you're getting this. It's not that he got only 4 plants. This is a ratio. So if he got 100 plants, 75% were tall, like one of the parents, and 25% were dwarf, like the other parent. Okay, so this ratio is important. It's called as the monohybrid ratio. So just to kind of summarize what we said here, because this is important. In a monohybrid cross, he took two parents of alternating forms. When he crossed it, he got the F1 generation. The F1 generation resembled only one of the two parents. From the F1, how did he get the F2? He did selfing, okay, or self pollination. And when he did selfing or self pollination to get the filial 2 generation, he noticed both the traits were expressed in a 3:1 ratio. So, this is what we have said so far. Now this will become clear when we look at an example. So here you can see he has taken a tall pea plant and he has crossed it with a dwarf pea plant. Okay, so now the first thing is to notice the phenotype. So phenotype is of this parent is tall, the phenotype of this parent is dwarf. Okay, just to revise, phenotype is the observable characteristic. Then the genotype, which gives rise to the phenotype. So the genotype of this is homozygous dominant because it's capital T, and this is homozygous recessive because it's small t. Okay, so he got the F1, which was capital T small t. Now, how did he get F1 plants which had the genotype of capital T small t? Because this plant is um, could produce only one type of gametes, that is bearing capital T. No other possibility is there here because the allele is only capital T. And over here, the possibility again only one type of gamete, small t. Okay, so when these two combine, okay, or they fuse, you get capital T, small t. Now, why are we writing only one allele in the gamete? Please keep in mind gametes are haploid. Okay, so that's why we're showing one allele here. So when they fuse, we know that your zygote is diploid and zygote will give rise to your plant. Okay, so the plant will bear capital T coming from one parent and small t coming from the other parent. So if this is the genotype of the plant, okay, what can we say about the genotype? Capital T, small t, it is heterozygous dominant. So the plant will be tall, but it is not homozygous tall, it is heterozygous tall. Okay, so all plants are uh, heterozygous tall in the F1 generation. This is the genotype, this is the phenotype of F1. Now coming to F2 generation, how did he get F2? We said selfing or self-pollination. That means TT is crossed with TT only. Okay, so now what are the types of gametes for a heterozygous tall plant? Two possibilities. Either the gamete has capital T or it has small t. Here again, either it has capital T or it has small t. So now what are the permutations and combinations or possibilities possible, okay, for the offsprings? So to do this, we use a method called as the Punnett square method. This is a Punnett square that is there. Okay, and all you have to do is to write the gametes of the parent on one side and of the other parent on the other side. So suppose this is the male parent, you're writing capital T and small t one side, and these are the two possibilities of gametes coming from this parent. So to fill this block, okay, you just have to write this T and this T. To fill this block, you will have to write this T and this T goes here and this T comes here. Then to fill this, you'll have this T and this T, and to fill this block, you have this T again and this T coming from here. So you can see you get four different types, or sorry, four individuals or four combinations possible. Here again are the combinations listed, the same four are listed over here. And as you can see that it is a 3 is to 1 ratio, 3 tall plants and 1 dwarf plant. So if I ask you what is the phenotypic ratio, it is 3 is to 1, 3 tall and 1 dwarf. But if you actually zoom in and see what is the genotypic ratio, you will see that 1 is homozygous tall, that is this plant. Two are heterozygous tall because they have two different alleles. Okay, so this is this proportion, and this one is dwarf, homozygous dwarf. Okay, so please note that the phenotypic ratio is 3 is to 1, but the genotypic ratio is 1 is to 2 is to 1. Okay, so this is important. Now, some more things to remember. Mendel proposed that the factors or the genes are the reasons that we have, similarities in our parents. So, inheritance is because of genes. At that time, he didn't call it genes, he called it as factors. Okay, so genes or factors today we know is the unit of inheritance which contains the information required to express traits. A gene, okay, is usually in pairs. Okay, so we say you have a gene pair, and in the gene pair you have two alleles. Like for example, this is a gene pair, you have two alleles here, T, capital T and capital T. Okay, so if both the alleles are similar, we call it as homozygous. So this is homozygous dominant pair, whereas this is, if the two alleles are different, we call it as heterozygous. This is already told to you. So this is heterozygous dominant, and this is same again. So two similarly, so homozygous recessive. Okay, so phenotype, genotype, homozygous, heterozygous is already explained to you. Dominant, recessive is already explained, so I won't waste time here. Coming back to the monohybrid cross that we did over here. Okay, so just to kind of revise that, the same thing in a monohybrid cross, he takes two parents with alternative forms of the trait. For example, here the phenotype is tall plant and dwarf plant. First, you write the genotype. The genotype here, it's a pure plant, so it is pure tall, and here it is homozygous dwarf. So these are two pure plants. In the F1 generation, you get T coming from here and T coming from here, because there's a gamete T and here there's a gamete small t. So you get all tall plants, 100% tall. But in F2 generation, what happens, which you get by selfing? So I said in the, this is your Punnett square here, you write, this is a mistake, gametes is a spelling mistake, GAMETES. You have capital T and small t here, and here you have the uh, other parent. So this, suppose this is the male parent and this is the female parent, then you write the female gametes this side, you write the male gametes this side. So what are the different possibilities? We work it out using a Punnett square. So I told you to fill each block, you just have to look at the adjacent block. So suppose you want to fill this block, you look at this and this. If you want to fill this block, you will look at this and this. You can even hold a ruler. Okay, so here you will look at this, and here you will look at this one and this one. So you get phenotypic ratio 3 is to 1, genotypic ratio 1 is to 2 is to 1. So Punnett square is what I showed you. It is in honor of the British scientist Reginald Punnett, and it is a very nice way to calculate the probability of the offsprings. So again, we have already seen that the ratio. Okay, so we won't waste time on this. In F1 it is 100%, in F2 it is 3/4 tall, 1/4 dwarf. So this again slide shows us the same thing. Then test cross. Now this is very interesting. Suppose you have a purple pea plant. Now from that chart there, I told you that if the flower color is purple, it is a dominant trait. Whereas if the plant has white flowers, it is considered to be a recessive trait. But as we know now, dominant could either be homozygous dominant or it could be heterozygous purple, right? So if it's a homozygous purple or heterozygous purple, we don't know the genotype. We only know that the phenotype is dominant purple. So how do we find out? It can easily be found out by crossing with a recessive parent. So here you can see, if you cross, okay, a purple plant which is, say for example, it is capital P, capital P, that is homozygous dominant, it is crossed with a white plant. So what will you get? How you'll write the gametes? You'll write capital P of this parent and small p of this parent. So you will notice that when you fill in the Punnett square, all four are plants. That means 100% of the plants are purple. Whereas if it was a heterozygous purple plant, you will get one capital P and one small p coming in the gamete. And when you fill in the Punnett square, this will be capital P, small p. This will be small p, again here small p, small p. So half of the offsprings will be purple, and the other 50% will be white. So if you get this result, okay, you can easily tell whether the parent purple plant was a homozygous plant or heterozygous plant by looking at the offsprings you get when it is crossed with a recessive parent. So this is called as a test cross. Okay, so finally in today's lesson, we'll just close by looking at the based on all these experiments, the first and the second law which Mendel formulated. So we have the first law, which is called as the law of dominance or the principle of dominance, and three important points to remember here. He said that characters are controlled by genes, but I told you he didn't call it as genes, he called them factors. and he said that factors occur in pairs, which we know is true today. So this was another thing that he talked about in his law of dominance. And the third thing that he spoke about in the law of dominance is that in a dissimilar pair of factors, one is dominant and the other is recessive. Dominant expresses itself and the recessive will hide itself. So these are the three important things to be remembered in law of dominance. Okay. Then he also formulated the second law, a principle called as law of segregation. Here it states that when a pair of factors for character are brought together in a hybrid, that means when you're making a hybrid, okay, we're using two different parent plants, the genes, they will not blend, they segregate and separate. And during gamete formation, there's no blending when the gametes are formed, but there is segregation and separation. Okay, so um, this is what he said. So this we will understand in a much better way when we take up dihybrid cross. Right now we have seen only monohybrid cross. Okay. Then another thing I'd like to close up with is incomplete dominance, because it is related to a monohybrid cross. Now it was noticed that in this plant called as the 4 o'clock plant or the Mirabilis jalapa plant, there is something called as incomplete dominance taking place. Okay. Um, so here you can see it's also seen in Snapdragon. So what was seen is, first we'll go to the example and then we'll come back to the note. So this is your dog flower snapdragon plant. It was noticed that there are two varieties. Okay, the red variety which is dominant and the white variety which is recessive. So red is a dominant, red flowers is a dominant trait, white flowers is the recessive trait. So as per Mendel's rules, what should have happened when red is crossed with white? In the F1 generation, it should have been all dominant, that means red flowered plants, but it was seen that the plants were pink in color. Okay, so showing you that red is not totally dominant in this plant, not in P. In this plant, that red is not totally dominant over white, okay, giving this expression of pink. So here what is happening is exactly this. When you cross a red dominant plant, capital R, with a white plant, small r, the F1 generation, which is a heterozygous, okay, it is a heterozygous dominant, capital R, small r, should have been red, okay, as per Mendel's laws, but in this case, the heterozygous plants were giving pink flowers. Okay, and when F2 was obtained, again the same 1 is to 2 is to 1 ratio was seen, but in this case you had 1, the capital R was red, the 2 R and small r were pink, and the 1 referred to the dwarf, which was small r. So what I'm trying to ask you to compare is this example with P. Now in P, capital T crossed with small t gave you all capital T, small t's, okay, a heterozygous plant which resembled one parent. But in incomplete dominance, what we see is when you cross a dominant with a recessive parent, you get incomplete dominance. So the expression of flowers here, flower color, is okay, is pink, because red is not totally dominant over white. Okay, so with this we'll stop up to here. Students, this would mean we have finished up till, okay, you will have to read up till 5.2.2.1, incomplete dominance. Okay, any questions regarding this same, please send me through WhatsApp. Thank you.